All right, membranes. What's bad about learning membranes from a book and pictures? Because they're dynamic. They actually move. And so we don't see that. So here are some experiments that actually told us that, hey, yeah, it does move. Okay, we're going to fuse two cells together, and we're going to photo bleach a cell. Okay. Here we take experiment 1, fusion. We take a human cell and we take a mouse cell. And what we can do is, okay, very slowly it's going to do it. Lee University's network is running fast. Okay, what we're going to do is label the individual proteins that are human proteins with a fluorescent label, in this case, red. And in the mouse case, they're going to label mouse proteins that are embedded in the membrane as green. Okay. Now, this is wonderful for men who are colorblind, because the biggest colorblind issue is red-green. And so I'm going to have to trust what you say when you see the next couple of slides, because I don't see it real well. Okay, so you see red on top and green on the bottom? Okay. And in experiments, we can create conditions in which we force the two cells to fuse. If by chance the membranes were static, then one half would be red and one half would be green, right? Over time. That is not what they found. Okay. If we allow the fusion to occur, okay, I think there's a video here. Please go. All right. At the top, you see red and green proteins begin to fuse. And then what happens to the distribution of the red and the green over time? They will distribute. The only way that they can distribute is if the membranes are free to allow things to move through. Proteins are pretty huge, and so if anything, proteins should stay, okay? But they're moving around. This also includes all of the membrane lipids can move around, okay? And so we can actually see this under a light microscope. Watch this movement, okay? Does one experiment prove anything? Does any experiments prove anything? No, we support the idea. So let's do another experiment. Okay. And so that other experiment, this is after several hours, is called FRAP. Okay. We're going to do photo bleaching. All right. And so what we're going to do is we're going to coat the membrane with a fluorescent chromophore. Oh, fluorescence. Do you know what fluorescence is? We use it all the time. We talk about it all the time. What is fluorescence? You might have even done it in the lab. I don't know. Do you actually understand what fluorescence is? What do you see when something fluoresces? You see light. Okay. But how is that light generated? Does it just glow all the time? No. Fluorescence is this. You shine a certain wavelength of light on the chromophore. All right. It absorbs that light. It gets excited. Can everything stay in an excited state for the entire time? No, it goes back to the ground state, and what it gives off is a wavelength of light that is different than the excitation. Okay, and so we can choose the color that we use because we can excite it with different wavelengths, and then what comes off depending upon what the molecule is. So we can get different colors, and so we have a whole lot of real viable look at that, and it's really a good technique. We can have things glow. Bioluminescence, okay, or we can chemiluminescence. We can use a chemical and treat it and actually glow. That's another way to detect. Or we could go all the way back old school and use radioactivity. We try not to use that too often anymore, but that's another way we can detect. Okay, we're going to take a membrane. We're going to cover it with fluorescent molecules. They're organic molecules. And then we're going to shoot it with a laser. Okay, and that laser is going to excite things, but give so much energy it breaks down the organic molecule. If you break down the organic molecule, does it fluoresce anymore? No, because it's gone. And so, let's try this.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light.
Ooh, this is real science fiction now. We shot it with our laser.
Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane.
Very mobile.
The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
So things can diffuse, but not everything diffuses. Okay? And so there are some things that are anchored, usually proteins, that might be anchored and organized over the surface where they're anchored. But there are other proteins, which the majority are, that actually can diffuse around, and we can observe that using this technique. Okay. Here is a example where we take a plant cell, we photo bleach it, and it will recover over time. Will we ever get back to the same intensity? No, we diffuse, and it will lose because we lose some of the colored material, but you will never be able to detect once it completely diffuses where that photo bleaching actually occurred. We can, or a graduate student can, watch this and calculate rates of diffusion over long periods of time, do lots of calculations, and we can look at different things and actually calculate how fast things diffuse. All right. Across layer migration really doesn't happen. We do have maybe one flip per hour in lipids, but it's very, very unlikely. We do have proteins that actually will, when they are synthesized, are placed into the membrane in a certain way. Maybe the N-terminal is always sticking on the inside, maybe the N-terminus is always sticking on the outside, but they are placed in the membrane in one particular way and they will not flip. So there is organization. Now, we did talk about a liposome, what this is, okay? What we would do is we would mix in a liquid solution some phospholipids on the top. And if you actually just pour the phospholipid on top, it'll be just like oil and water. It'll float on the top, and the water will be on the bottom, and it doesn't mix very well. But if you shake it up really well, and really we don't really shake it, what we do is we use sonication, sound waves. That really does a very good mixing. Depending upon how intensely the sound waves and how long the sound waves are, we will create small envelopes called liposomes. And it is essentially a lipid bilayer with a really tiny compartment on the inside. Now that liquid, the water that we place inside, or the buffer system that we place inside, we can manipulate what's in there. We can manipulate what's dissolved. And so when we create liposomes, we trap a little bit of what's in that sample. And so we now have a delivery system to take something on the inside of that liposome, fuse it to the membrane of something else, and allow delivery in a experimental capacity. And this kind of delivery, we can also put in proteins that would be targeting molecules that would bind maybe to a certain type of cell structure. And so we could put the drug in the liquid, we could put the protein in there and the lipid, mix it all up, create liposomes, and then use those liposomes to target individual cells in which we want to deliver that drug. This is an experimental procedure that is used, and it's a little bit of art, a little bit of science to create a delivery system. Now, the cells themselves have proteins on the inside, on the surface, and there are a class of molecules called cell adhesion molecules. All right. If we just had a cell bound to, next to a cell, all right, and you grow them up in the lab, there's not a lot of attachment to them. But let's say the cells in your body, there's some integrity between the cells that hold things together. And what happens is, it is the proteins that are embedded in the membranes that make associations to other proteins that hold things together. They're all classified as cell adhesion molecules. Okay? And so the cases that we have or the different classes are called, first is integrins. They are proteins that have, they're heterodimeric. Okay, what does that mean? They are two proteins put together, and they're two different proteins put together. We call them A and B, or alpha and beta. Okay? They come together, and they serve as binders, okay, to other proteins. And a lot of times, integrins can bind to themselves. Cohedrins, on the other hand, are homophilic proteins that interact with identical cohedrins in other cells. So I've got one cohedron here and one cohedron here, and they bind to themselves, but they bind across between and hold two cells together. That binding is facilitated by calcium being in the mix. If calcium's there, they bind. If calcium's not there, they do not bind. There's a whole class of proteins that have immunoglobulin-like properties. What's an immunoglobulin? Do you know what that is? It's a big word, right? Yes. Antibody. Okay, what does an antibody do? Just in general. What is significant about an antibody? It binds antigens. Okay? It binds super tight to something. The antigen is what it binds to. This class are proteins on the surface of membranes that have binding pockets that are very strong and very specific. Okay? That bind together. And that allows us to have adhesion, or binding together. Okay? The last class of cell adhesion molecules are what are called selectins. We'll come back to a selectin in a little bit, not in this chapter, but a couple chapters. A selectin is this. On the outside of a cell, I have a binding protein, and what it binds to is a sugar, okay, or a group of sugars. Now, a lot of other cells will present on the outside sugars. I told you sugars are presented on the outside. Well, the selectins will be searching for that sugar so that the sugar that is bound to the membrane of a cell and this cell bind together. They also do it in a calcium-dependent manner. Calcium's there, they bind really good. Calcium's not there, they don't bind very much at all. These are ways in which we hold cells together, the cell membranes together, to have integrity in our tissue types. Now, membranes will fuse, and we've shown that. But when we have membrane fusion, there's a whole bunch of things that it's required. We usually have a receptor-mediated event, meaning that on one cell, there's something that presents out, and another cell, there's something that presents out, and they have to bind together. That binding together brings them close. It also causes changes in the protein of the binding proteins that cause internal structural changes that bring the two even closer together. We have to get rid of water and substitute that. We have to disrupt the surface of both of the membranes so that the lipids like to meet and they will become a single bilayer through that fusion event. Okay? We see that. Let me show you a particular process in which that fusion occurs. Okay? You've probably heard of HIV. Okay? HIV is a virus, and the virus has to fuse into your cells, in particular into T cells. Okay? But here's where the bad part comes out. The virus has on its surface a protein sticking out as a certain shape that binds to receptors that are only on T cells. Okay? And so it's presenting something that the body would normally present, but it's a virus. So it's giving the wrong address. And so the protein, in terms of GP41 and GP20, acronyms, I hate them. Okay, it's called a glycoprotein 41. Glyco meaning it's a sugar protein, a protein that has sugars on it, that is 41 kilodaltons large. Its weight, that's how they found it. And glycoprotein 120 is a protein that has sugars on it that weighs 120 kilodaltons, or 120,000 grams per mole. Right? And so it's presenting these proteins. It binds to a receptor called CD4, and they bind well together. That causes a conformational change in which a protein itself gets incorporated, the GP20 gets incorporated into the membrane of the T cell. Now the virus cell wall and the T cell wall is only now a protein away. Okay? And when the protein GP20 gets on the inside, see those little pieces of protein that are on the inside? They start interacting with what's inside, causing a structural change in that protein, which brings them close together. Let me show you what that looks like, actually. Here's the mediated event. I'm sorry, it was GP41, not 120, that actually made the binding. Here is on the top the virus, and on the bottom is the T cell. It's already been incorporated. But those two cells are really close, but not close enough to fuse. What happens? Okay. We see the proteins, they're stuck inside. And what happens, once the ends of those proteins feel the inside of the cell, something interacts with it on the inside, causing the protein to change shape, to change its structure. Okay, so we've got long organizational structure. We'll talk about these structures next time, what they are. You probably have heard about an alpha helix. But do you see how large a structural change that occurs? It goes from something that is random into a big change into an alpha helix. But what really happens is there's a consequence when that structural change starts to interplay. When I actually have that. Come on, show it to me. There it goes. As it makes the structural change, you see the membranes are coming together, and it actually buds out the two membranes, and it creates the local disruption. And in every fusion, almost every fusion, there is something similar. It has to be physically moved to bring those together. They don't just randomly come together. You actually have to force it to come together. Pulling it apart, creating a structural instability to make those membranes fuse. And now the membranes are fused, and I have a polar region in which the cytosols can mix. Okay, it doesn't matter that this is HIV. I just found a good animation with HIV. All membrane fusions have similar structures in that proteins will interact to bring those membranes together. Okay, it is like two balloons. You can't take and throw balloons at each other fast enough for them to fuse. Okay, you actually have to physically do something to disrupt the membrane, and proteins will serve in that capacity.